Hey everyone, and welcome to this explainer. I am absolutely thrilled to be your guide today as we jump into what is honestly a fascinating, completely invisible world. Think about it for a second. We know the cell is the fundamental building block of literally all living things, right? It's the engine of life itself. But there's a massive catch. To the naked eye, cells are basically completely invisible. So how do scientists actually investigate a crime scene they can't even see? Well, today we're taking a grand tour of the cell detective's toolbox. We're going to look at the exact methods, the ingenious tools, and the pretty wild processes researchers use to crack open the microscopic world.
So here's our lab manual for today. We'll start with Part 1, how to see a cell, then move to Part 2, light versus electron microscopes. Part 3, prepping the perfect sample. Part 4 is lighting up the cell. And we'll wrap up with Part 5, breaking cells apart. Let's dive right in.
All right, Section 1, how to see a cell and the limits of the naked eye. To breach that microscopic barrier, we essentially have to overcome the limitations of human biology just to study human biology. And, a really common misconception about microscopes is that they're all about magnification, just making things bigger. But honestly, simply magnifying a blurry image, that just gives you a really big blurry image. The actual secret sauce here is something called resolving power. This is the ability of a microscope to look at two microscopic points that are incredibly close together and still distinguish them as two completely separate, fine details, rather than just one merged, muddy blob. And resolving power is heavily dependent on the wavelength of whatever beam you're using to look at the sample.
Moving on to Section 2, light versus electron microscopes, a tale of two lenses.
Okay, so this is where we compare the traditional method of bouncing visible light off an object with the super high-tech method of shooting an actual beam of electrons at it. Optical microscopy, which is kind of like what you probably used in high school biology, uses visible light. But because visible light has relatively long waves, its resolving power hits a literal theoretical brick wall at 0.2 micrometers. Its maximum useful magnification tops out at about 1,000 times. But electron microscopy, oh man, it uses an electron beam, and electrons behave like ultra-short waves. Because these waves are so incredibly tiny, they slip between much smaller details, giving us a resolving power of 0.2 nanometers and a staggering maximum magnification of 100,000 times.
The crucial difference really boils down to this mind-bending number, 1000x. An electron microscope provides a resolving power that is 1,000 times higher than a conventional optical microscope. To put that into perspective, it's literally the difference between looking at a giant paper map of a whole city and being able to zoom in enough to see a single pebble sitting on the sidewalk.
Now, there are two main flavors of this electron technology. First up is the transmission electron microscope, or TEM. In this method, electrons are fired completely through an ultra-thin slice of a cell. The denser parts block the electrons, casting these internal shadows that let us see the ultrastructure and the organelles inside, kind of like a medical X-ray. Then there's the scanning electron microscope, the SEM. Here, the beam doesn't pass through at all. Instead, it scans the surface of a sample line by line, bouncing off to give us these absolutely stunning, highly detailed 3D topographies of the outside of the cell.
Which brings us to Section 3, prepping the perfect sample, the delicate art of slicing the evidence. So you can't just grab a piece of raw tissue and stick it straight under a high-powered lens. It doesn't work that way. Let's walk through the really rigorous step-by-step procedure of standard sample prep for an optical microscope. Step 1 is fixation. We basically have to freeze biological time. We use chemicals like formalin or Boone's solution to instantly stop all cellular activity and preserve the structure exactly as it was in life. Step 2, dehydration. We carefully pull all the water out by putting the tissue through baths of stronger and stronger alcohol. Step 3 is embedding. We infiltrate the whole thing with molten paraffin wax, which hardens it into a solid, sturdy block. And finally, Step 4 is sectioning. We use this machine called a microtome, which honestly is essentially a microscopic deli slicer, to cut ribbons of tissue that are just 2 to 5 micrometers thick.
But wait, we aren't quite done yet. A 2-micrometer slice of tissue is virtually transparent. You can't see anything. To actually spot the cellular structures, we use chemical stands to force them to reveal themselves in brilliant colors. H&ES is a classic. It uses different chemical affinities to dye the DNA, a basic blue-purple, and the acidic cytoplasm pink. Algin blue specifically targets and reveals mucus hidden within the tissues, and PAS highlights carbohydrates, turning them a vibrant red or purple, which is super useful for spotting things like glycogen stores. We're quite literally painting the invisible so our eyes can see it.
Now, the prep is even more extreme if you're using a scanning electron microscope. Because the SEM blasts the sample with an intense beam of electrons, the prep relies on this hardcore freeze-fracture method. First, cryofracture. Scientists rapidly freeze the tissue and then literally physically crack it open to expose the internal cellular landscape. Second, sublimation. They toss it in a vacuum to essentially suck out the water ice without actually melting it. And finally, metallization. They deposit a microscopic coating of heavy metal, often real actual gold, over the entire sample. This conductive gold layer stops the sample from burning up under the intense electron beam and bounces the electrons back to the detector to create that gorgeous 3D image.
Up next, Section 4, lighting up the cell, making the suspects glow in the dark.
Okay, sometimes we aren't just looking for general structures. We are looking for one highly specific protein hiding in the dark amongst millions of others. To find it, we use fluorescence microscopy. The physics of how this works is brilliantly simple, yet profoundly powerful. It happens in 3 steps. First, excitation. We tag our target protein with a fluorescent molecule called a fluorochrome. Then we zap the sample with a very specific high-energy wavelength of light. The fluorochrome absorbs this energy, heavily exciting its electrons. Second, emission. Those electrons quickly drop back down to their normal state, and when they do, they release all that excess energy as a new, longer wavelength of light. Basically, they glow. Third, optical filtering. We use special filters on the microscope to completely block out the original laser we fired at it, allowing us to see absolutely nothing but the pure, glowing light emitted directly from our specific target protein against a pitch-black background.
Let's look at our roster of glowing molecular tags, because scientists have an entire palette to work with here. Fluorescent, or FITC, glows a brilliant, bright green and is heavily used to detect specific proteins. Rhodamine, TRTIC, glows a fiery orange-red and is absolutely perfect for highlighting cell membranes and the structural cytoskeleton. DAPI is this remarkable dye that specifically binds to DNA, glowing bright blue. It's the absolute go-to tool if a scientist needs to count how many cell nuclei are in a sample. And GFP? That is a total game changer. It's a naturally occurring green fluorescent protein originally found in jellyfish, and scientists can actually genetically program cells to produce it all by themselves.
All right, our last stop, Section 5, breaking cells apart, the highly calibrated blender.
So far, we've really focused on visual tools, but sometimes just looking isn't enough. To truly understand what a cell is made of, sometimes we have to just dismantle it completely through a process called homogenization. Honestly, think of it as putting the cell in a highly calibrated blender. But it's a super delicate balancing act. We need to completely destroy the tough outer plasma membrane to release the contents, but, and this is crucial, we absolutely must not damage the incredibly fragile microscopic organelles floating inside. To pull this off, scientists grind the cells in a very specific bath, an isotonic 0.25 molar sucrose solution. This sugary liquid perfectly mimics the internal pressure of the cell itself, so when the outer walls come down, the inner components don't just burst from osmotic shock. Once we have the cellular soup, which is called the homogenate, we have to sort the parts out. We do this using a centrifuge to spin the liquid at incredibly high speeds. Notice how increasing the spinning speed pulls different organelles down to the bottom of the tube to form a solid pellet, strictly based on their mass. At a low speed, the heaviest components drop out right away, the heavy nuclei and any unbroken cells. We pour off the remaining liquid, called the supernatant, and spin it again, this time at an intermediate speed. This forces down the medium-weight organelles, like your energy-producing mitochondria. We pour the liquid off one more time and spin it at an extremely high speed to pull down the very tiniest elements, like the protein-building ribosomes.
Now, differential centrifugation is a fantastic, standard tool, but it brings up a really critical question, one that totally frustrated early cell biologists. What if the organelles weigh exactly the same? Because, as it turns out, mitochondria, lysosomes, and peroxisomes all sediment at almost the exact same intermediate speed. If you try to separate them by mass alone, you just end up with a mixed-up pellet. And that brings us to the ultimate solution, density gradient ultracentrifugation. Instead of a uniform liquid, scientists create a column of liquid in the tube where the density slowly increases from the top to the bottom, usually by using varying concentrations of sucrose. When you put the cellular soup on top of this and spin it, each organelle is forced down through the gradient until it reaches the exact zone where the liquid's density perfectly matches its own. It essentially acts like a microscopic biological buoy, floating precisely in its specific density zone. This allows us to flawlessly separate mitochondria from lysosomes, even though they weigh the exact same amount. We've covered some truly incredible ground today, looking at optical limits, electron beams, freezing, slicing, glowing jellyfish dyes, and ultracentrifuges. But why go through all of this? Why spend decades developing these wildly intense, meticulous methods? Well, because as our source material beautifully states, the cell is both the elementary and functional unit of all living organisms. To understand the cell is to understand the very fabric of life, health, and disease itself.
Which brings us to the end of our explainer and leaves us with a pretty provocative thought. By continuing to push the limits of our technology, evolving from those simple glass lenses of the past, all the way to today's nanometer-precise electron beams and fluorescent markers, what invisible biological mysteries are we going to crack open next? The Cell Detective's toolbox is always growing, and the next big discovery is just waiting out there to be brought into focus. Thank you so much for joining me on this deep dive into the microscopic world. Keep your curiosity alive and keep exploring.